Okay, and great. So let me back up here. So today I would like to welcome you all. We've got a mix of students, staff, faculty, and community members here, and we're so thrilled that you're joining us for this month's Let's See You Well presentation. So a little bit of background, the Let's See You Well speaker series allows arts and sciences researchers and practitioners to interpret the 8 dimensions of wellness from their perspective, thus articulating the value of a liberal arts education through the lens of wellness and overall health. The Let's See You Well experts of the month provide wellness content for lectures, participant prompts, and Be Well promotional materials, and are encouraged to articulate wellness through the lens of their research, scholarship, and creative practice, while holistically and comprehensively showing the interrelatedness, interconnectedness, and essentiality of health and wellness across our many life forms, including plants, animals, and human animals, and the environments in which they reside, land, water, and air. We are fortunate to have faculty who study this multifaceted planet on which we live to optimize health and wellness across all life forms and environments. Today, I am so thrilled to host our September Let's See You Well expert, Dr. Deane Bowers, professor of ecology and evolutionary biology, and the CU Museum Curator of Entomology. Professor Bowers has served on the CU Boulder faculty since 1989 and previously served on the faculty of Harvard University. She holds a PhD from the University of Massachusetts Amherst. In 2015, she was named the Entomological Society of America Fellow, and in 1996, she won the Boulder Faculty Assembly's Award for Excellence in Research, Scholarly, and Creative Work. Again, this workshop will be recorded, and closed captioning is available by clicking the CC button to view the live transcription. We do plan on having time for questions at the end of the session, so please hold your questions, and I'll be sure and monitor the chat and raised hands. And without further ado, I am thrilled to introduce Dr. Deane Bowers and her presentation titled Globalization's Consequences for Plant-Insect Interactions and the Planet's Wellness. I'll turn it over to you. Welcome.
Hi, everybody. Thank you so much for coming today. And let me share my screen here. Let's see. I'm not getting a prompt for doing that.
Do you have the green button highlighted on the panel below?
Yep. Okay. Okay. All right. Can everybody see that?
Looking good.
All right. So what I'd like to do today is talk about how increased movement of humans across the planet has really changed the environment in which we live. And I'm trying to do this in the context of the CU Be Well program. Now, I was a little concerned about that in the beginning, but we'll see how it goes. And so I'm putting this into the context of the sort of 8th dimension of wellness, which is the environment. And a lot of the research that goes on in my lab deals with how we as humans interact with our environment, and especially in the context of insects and the plants that they feed on. So globalization has affected not only humans, but it's made the planet get smaller, not physically smaller, but smaller in terms of how we can access so many different parts of the globe. And as we increase the rate at which we do this, we're moving not only ourselves around the planet, but we're also moving a lot of other organisms around the planet. And although this is a phenomenon that's been going on for millennia, this is something that has increased hugely in the last few decades. And as we move things around the planet, you can see here, these are shipping journeys over in 2007. And so this just shows how much we're moving things around, moving ourselves and other things as well. And as we move across the planet, not only do we move, but we take a lot of different kinds of organisms with us. And what I'm going to concentrate on today is the movement of insects and the plants on which they feed across the planet, and how this movement might affect native insects in the areas to which these plants and insects go. And ultimately, that movement can then have an effect on humans and on our environment. So as I mentioned, I'm going to talk about insects and plants, and what I'm showing you here is the pie of life. So this shows the relative number of species of all these different groups of organisms. So you can see chordates, the group to which we belong, is a really teeny little slice of this pie. But what I'm going to be talking about today are insects, which are a really large slice of this pie. More than half of the known species on the planet are insects. And here are plants, a smaller component, but also extremely important. And for insects, about half of all known insects feed on plants. And so there's about 1.3 or 1.4 million species of insects known, and about half of those are plant feeders. And when I talk about plant feeders, I mean not only insects that chew on leaves, but also insects that eat other parts of plants. For example, pollinators consume nectar and pollen from plants. So I'm going to be talking about plant feeding insects and the plants that they feed on. So you hear a variety of different terms used to refer to organisms that have moved from their place of origin into a new place, a place where they haven't previously occurred. And you see the term introduced, you see the term exotic. And when organisms start moving around and getting in the way of humans or negatively impacting human welfare, they're called invasive. So you can see all three terms, and these are terms that humans have applied. And the negative connotation of invasive is something that we humans have put on these organisms, but because it means they're getting in our way in some way. And so an introduced organism isn't necessarily an invasive organism. So I'd like to start by talking a little bit about exotic plants, and then I'll talk about exotic insects, and then I'll talk a little bit more about some of the research in my lab that deals with how moving plants into novel environments can affect the interactions of the insects that occur in those new environments. So a couple of examples of exotic plants. The first one is Dyer's woad, Isatis tinctoria in the cabbage family. And this is a dye plant that produces a beautiful blue color like indigo. It's a different plant species from indigo, but it was brought into the US in the 1700s for its dye potential. And any of you who are Braveheart fans may be familiar with it in this context. It was also used in Europe as a body paint, making this beautiful blue color on the skin as well. And so this was brought over for its utilitarian purpose of dyeing clothing. However, this has spread dramatically, and here in the West, it's not so bad in Colorado, but in other places in the West, this has become a serious problem. This is a field that's been taken over by Dyer's woad. Another species that's considered invasive, that is very common here in Boulder, is Dalmatian toadflax, Linaria dalmatica. As you can see by the flowers, it's a really beautiful plant. It's closely related to snapdragons. And so it was introduced in the early 1900s as an ornamental. And that too has
Deanne, I'm so sorry, or Deane, I'm so sorry I muted everyone. Would you mind unmuting yourself again, just to,
Okay,
thank
can
you.
you hear me now?
Yep, sounds good, thank you.
Okay, great. Okay, so could everybody hear me with about Dalmatian toadflax? Was that part? Okay, great. So, so these kinds of invasive plants have been a real problem here in Colorado. So they can spread very quickly. Here's Dyer's woad and Dalmatian toadflax. You can see annually they're spreading by 14 or 8% a year. And this has serious impacts on grazing, which here in Colorado is particularly important. So these plants were introduced for initially valuable reasons as a dye or as an ornamental, and they've become a problem. What about some exotic insects? Well, again, here in Boulder, this is one probably well known to many of you, the emerald ash borer. So this is a beautiful beetle you can see here in the family Buprestidae, whose larvae, and here's the larva right here, the larvae bore into wood and ultimately kill the tree. So it was introduced into the east coast of the US in 2002. And in September 2013, it was first found in Boulder. So a really fast spread, and it's devastating to ash trees. And just to show you the impacts here in Boulder, this is a picture actually from here in Boulder. This is a tree lined with ash that has all been killed by emerald ash borer. And ash, especially white ash and green ash, are planted extensively across the United States as ornamentals and street trees because they're really beautiful, they're easy to grow, and they grow quickly. And so this is what had to happen to all of those trees. They have to be taken down. And so emerald ash borer is a serious problem here in Colorado, as well as across the country. Ash is the host plant for over 300 species of insects. Some of these feed native insects, and some of these feed, it's a native tree, some of these feed exclusively on ash. And so if ash disappears, we're also going to be losing those native insects. And then here's one of my favorites, even though it's a really serious problem, the spotted lanternfly. Any of you who've spent any time on the East Coast, especially New Jersey and Pennsylvania, may have encountered this creature. They're super cool looking. They're about 1 inch to 1.5 inches long. This is an adult, and this is a little nymph. They probably first arrived in the US in 2012, but the first infestation wasn't discovered until 2014, and they are wild looking. So their hind wings have this really bright color. And I was in New Jersey last fall, so I got to experience them firsthand. And I sort of freaked out a waiter when I one landed on our table at a restaurant, and I reached over and picked it up to show it to my sister. And the waiter was like, "Oh my God." But they're super cool, but they are a very, very serious problem. So they feed and do serious damage to a lot of important crop plants, especially fruits and vegetables. And here's a mass of them on grape. And here is a tree trunk covered by the adults. So they're a very serious problem, and they're moving very quickly. I love this slide, which really depicts the way people on the East Coast are thinking about this species, because the invasion happened really fast and the numbers are huge. And so this is a map that was updated just a week or so ago, August 24th. And initially, the first infestations were in New Jersey and Pennsylvania. And so now you can see they're up into central Massachusetts, upstate New York, and even over here into Ohio. So it's moving really, really quickly. Fortunately, modeling has suggested that because of the conditions much further west than this, it's not going to be able to spread into Colorado. But one of its favorite host plants, the tree of heaven, is a weedy invasive tree species that occurs commonly here in Colorado, so it's not impossible. This is a very serious invasive species. Okay, so I've given you some horror stories about exotic organisms that have become invasive organisms, but I want to give you a little bit of a different view about exotic plants and insects. And first is about our food. So this is a map showing the areas of origin of the fruits and vegetables that we eat here in the United States. And so these include, all different kinds of fruits and vegetables. You may not be able to see the details, but you can see that there's lots of different icons here. And one of the things that you might notice right away is that we here in North America are pretty depauperate in the fruits and vegetables that are native to North America that we eat. And many of these that come from other places are now grown here in North America. But there's only 6 fruits and vegetables that are native to North America. So we have brought all these exotic fruits and vegetables into North America. We raise them here, and they're a critical component of our diet. So our food, most of our fruits and vegetables are exotic organisms. Do we consider them invasive when we see fields of corn or orchards full of oranges and grapefruits? I don't think so. Also, many of our prized garden plants are exotics. Daffodils are native to North Africa and Southern Europe. Tulips are native to Central Asia. So many of the ornamentals that we put in our gardens and love to grow come from other places, but they're not considered problems until they start interfering with things that are important to us, such as overtaking rangelands. So what about exotic insects that aren't so bad? Well, of course, the first one that comes to mind are honeybees. Most of you probably know, but some of you may not, that honeybees are not native to North America. They could be considered an invasive species. They were brought over by the early settlers. They are cultivated and raised the same way that cattle are cultivated and raised. We keep them in hives, and they perform pollination services on many of our crops. And they produce important things like honey and beeswax that we use. But it's important to remember that honeybees are cultivated. They're not native to North America. They interact occasionally in negative ways with some of our native bee species. Occasionally, we do have what are called feral colonies of honeybees, although these are becoming less common. This is actually a feral colony. This picture was taken at White Rocks, which is right outside Boulder, several years ago. And you can see this is what the combs look like in nature. And this is a picture I just love. This is a hive that was formed underneath a barbecue grill. Somebody had covered it up, hadn't used it in a year. The honeybees found it and made this beautiful nest. So how important are honeybees? And I want to say here that people think of honeybees as being the main pollinators of many of our crops, but that in fact is not the case. They may be the main pollinators, but they're not the sole pollinators. Many of our native bee species are very important crop pollinators. And how important are these? They're really important. They're worth a lot of money. So it's been estimated that honeybees contribute about $20 billion in services, specifically in pollination services, to US agriculture. But other bee species, especially bumblebees, but other bee species as well, can contribute about half that much. So we're talking $30 billion here, and that's probably an underestimate. And to give you an idea of the importance, I love these pictures. This is what our produce choices with bees are like. the way things are now. And this is what it could be like without bees. So we're missing a huge number of fruits and vegetables that depend on bee pollination for successful fruit production. Okay, so now so I've given you some examples of the wonderfulness and the difficulties of some of these exotic or invasive species. So now what I'd like to do is take a little bit of a different tack and talk about what happens when novel plants are introduced into native ecosystems and start interacting with the organisms that exist in those novel ecosystems. And to do that, I'm going to talk about a plant that my lab and others have done a lot of work on. And this is probably a plant that's familiar to most of you because it occurs all over Boulder, although you may not have noticed it. It's a common weed here in Boulder. It's called English plantain, or scientific name is Plantago lanceolata. And it's producing these flowering stalks right now. And it has many different names. You can see English plantain, ribwort plantain, all of these. My favorite, though, is white man's footsteps. And this name comes from the fact that it was introduced into North America about 200 years ago. And it has these really sticky mucilaginous seeds that stick to everything. And so this species was spread across North America with the early settlers because these sticky seeds stuck to the shoes of the settlers as they moved west and the hooves of the animals that they took with them. Hence the name white man's footsteps. And it turns out that in other cultures, they have names that are similar to this that refer to the fact that this species spreads by sticking to human feet and being spread across the landscape. So as I said, it was introduced into North America about 200 years ago, and it's extremely successful. It's spread all over the country. This species is considered invasive in 4 states in the US, but even though it's really common in all other states, it's not considered invasive in any other states. And here in Colorado, it's not considered invasive. But it's been introduced into all continents. It's native to Europe, and it's been introduced into all continents in the world except for Antarctica. So it's really widespread. In New Zealand, it's cultivated as a forage crop for cattle and sheep. So this is a field of Plantago lanceolata, narrow-leaved plantain. So it's a really common plant across the globe. So as I mentioned, I'm really interested in what happens when these introduced plants get incorporated into the diets of native North American or native herbivores in their exotic homes. And I work primarily on butterflies and moths. And this plant species has been incorporated into the diet of many native North American herbivores, probably at least 100 different species that had never been exposed to this plant species before now feed on it. And these are some of the butterfly and moth species that we've worked on in my lab. You can see a lot of them are really beautiful. But all of these different species now commonly feed on this introduced plant, even though it's only been here for a couple of 100 years. And one characteristic that's important in the consequences of this plant for native herbivores are the chemical compounds that it contains. And so one of the things that my lab is interested in is how the chemical compounds in plants can affect their interaction with the insects that they feed on. And this plant contains a group of compounds called iridoid glycosides. Now, you may think you've never heard of iridoid glycosides, and maybe you haven't, but it's likely that you've experienced them in some way. So they're very widespread in plants. If you've eaten blueberries or cranberries, you've eaten iridoid glycosides. They contain iridoid glycosides. This is penstemon, a common ornamental and also a native plant here, Indian paintbrush, honeysuckle, catalpa. These are plants that all contain iridoid glycosides. These compounds are extremely bitter, but they're also important medicinally to humans. For example, if you've ever taken valerian for insomnia, the active ingredient in valerian is iridoid glycosides. If you love or hate olives, it's probably due to iridoid glycosides. They're responsible for the bitter taste of olives. If you've sadly had to take Metamucil, you've experienced iridoid glycosides. And more recently, the antioxidant properties of iridoid glycosides are being taken advantage of, and you can live younger longer if you drink some of these drinks that contain fruits that contain iridoid glycosides. So these are compounds that are really important to humans, but they're going to play a role in what I'm going to be talking to you about today with regard to English plantain. So from the insect perspective, what happens to these chemical compounds in plants when they're ingested by insects? And this is one of the species that actually has the ability to store these iridoid glycosides. And so when an insect eats these chemical compounds, when it eats a plant, they can just get rid of them. They can excrete them in their fecal material, or they can break down or metabolize them in some way. But I'm especially interested in insects that have the ability to sequester these compounds, to store them in their own bodies, so they retain them in their own bodies. And that can make these insects taste disgusting to potential predators. And that's what's going on in this species. It's very brightly or warningly colored to advertise the fact that it tastes really bad. And work in my lab and that of others has shown that when an insect can sequester iridoid glycosides, it makes them taste really terrible. And so we've worked with spiders, ants, mantids, some bird taxa, wasps, and all of these avoid insects that have the ability to sequester or store these compounds. This is a similar story to the monarch butterfly, which stores cardiac glycosides from its milkweed host plants, but this is a different group of compounds and a different set of plants, but it's a very similar situation. So, as I mentioned, this plant has been incorporated into the diet of many native North American herbivores, and I'd like to talk about work with two of them. With this species, the white peacock, and with this species, Taylor's checker spot. And I'll talk about Taylor's checker spot first. And so this is a story about rare species conservation. So it turns out that Taylor's checker spot, Euphydrius editha taylori, is an endangered species. And here's a gorgeous picture of the adult, and here are the caterpillars. And this species' primary native host plants are these two species of Indian paintbrush. One of its host plant species is actually also endangered. That sounds like a tough life, right? Castanea levisecta. But another species, Castanea hispidus, is not endangered. And we have several different species of Indian paintbrush, as it's called here in Colorado as well. And so called because it was an important dye plant for Native Americans. And this species is native to the Pacific Northwest. This slide shows its original range, its historical range, but now there are only a few, 3 or 4 populations of this butterfly that are remaining. And so there's some pretty major conservation efforts trying to keep this butterfly from going extinct. And one of the things that was discovered in these efforts is that this species has started feeding on English plantain. And so it was found in the field. So in these native habitats, English plantain occurred, and it was found that caterpillars would feed on this plant and females would lay their eggs on it. So the questions that we were interested in were, what are the consequences for this endangered species for feeding on this introduced host plant? And this was a project that was done with Nate Hahn, a graduate student, and his advisor, John Backer, at the University of Washington in Seattle. And so we wanted to look at what the consequences of feeding on this introduced plant were for this endangered species. And I'm not going to go into the details of the data here, but I want to tell you first, we found that females would happily lay eggs on it, and they would lay eggs on it at the same rate at which they would lay eggs on the native species, on the Indian paintbrush species. And we found that caterpillars grow well on it, and they grow as well on this introduced species as on one of the native host plants. So it's looking like this may not be such a bad thing for this butterfly species, because one consequence could be that, you know, females might lay eggs on it, but the caterpillars might not be willing to feed on it. But in fact, what we found is that females will lay eggs and caterpillars will grow well on it. But given my interest in chemical defense, we also wanted to know, how does feeding on this novel host plant affect levels of chemical defense? So these Indian paintbrush species also contain iridoid glycosides. And so how does the ability of caterpillars to store chemical compounds and the levels of these iridoid glycosides that are stored by these caterpillars compare on the native host plants and on the introduced host plant? And what we found was that here's one of the native host plants here, and here is the introduced plant, that caterpillars are sequestering about 5% dry weight of these compounds. And there's no difference in this Castanea hispida and the introduced plant. But the second native host plant, this is actually the endangered one, caterpillars were really poorly chemically defended. So caterpillars that are feeding on this native host plant could be relatively palatable to potential predators, whereas if they're feeding on the introduced plant or the other native species, they could be quite unpalatable. So it doesn't look like this plant is having negative effects on this native species. So this introduced plant can be a good thing for this endangered species. It also turns out that in the areas where Taylor's checker spot occurs, these Indian paintbrush plants come out in the spring and the caterpillars start feeding on them. At the same time, they're feeding on the English plantain. But as the summer progresses, the Indian paintbrush dry up and senesce. And as it happens, they senesce. When they senesce, there can still be English plantain around for the caterpillars to feed on. So this can be another resource even in years when these native plants aren't available, because they stay green and yummy for longer than the native host plants do. Okay, so now I want to talk about a little bit different kind of story that doesn't involve an endangered species, but involves a species that's native to the southern US, this butterfly here, the white peacock. So the white peacock, or Anardia jatrophy, is a species found in the southern US. It's particularly abundant in Florida, and I'm from Florida, so this place has a particularly warm place in my heart. And it's fairly common in southern Florida. And its primary host plant in Florida is water hyssop, Bacopa monieri. And if any of you are aquarium lovers, you may have experienced this plant species. It's planted a lot in aquaria. It likes really wet, moist habitats, but it does not contain iridoid glycosides. So in 2010 was the first recorded incidence of caterpillars of this species feeding on English plantain. And so we were really interested in what the consequences of feeding on this novel host plant were for this native Florida butterfly. So we reared caterpillars on the native host water hyssop and on English plantain. And we wanted to see what the consequences were. Oops. So we found first that caterpillar growth on this introduced plant was slower. So this is the number of days that it took for the caterpillars to pupate, to reach the pupal stage. And it took about 20 days on the introduced plant, but only about 16 days on the native plant. So it takes quite a bit longer to develop. And that can be a bad thing because that means that the caterpillars can be out in the environment and available as food for potential predators or as a resource for pathogens. So slow growth can be a bad thing. But we also found that the pupal weight, the weight of the pupal stage was quite a bit higher, about 20% higher on the introduced plant than on the native plant. And a bigger pupa means a bigger adult, and bigger adults can produce more offspring. So this greater pupal weight can be a good thing. So it may take longer to get to the pupal stage, but when they get there, they weigh more. So we also found out that when these caterpillars eat English plantain, they sequester iridoid glycosides. So they become chemically defended when they're feeding on this novel host plant. So these are 4th instar caterpillars, 5th instar caterpillars. They keep a little bit into the pupal stage, and they even keep a teeny bit into the adult stage. So this novel host plant then can result in caterpillars becoming unpalatable. But some of our more recent work has been dealing not with predators, but with pathogens. And so we've been working on a viral pathogen that affects caterpillars. It's a fairly widespread pathogen. It's called Junonia senia densovirus. That's because it was first isolated from a butterfly species, actually, Junonia senia. And what it does is it turns caterpillars to mush. However, so this is a poor caterpillar that's been fed on a leaf that had this virus on it, and it's just turned to a blob of mush. And so this virus can kill caterpillars. However, the response of caterpillars is dose dependent. So if they get a low dose of this virus, they can survive. But at high doses, it can kill them. So we were interested in how feeding on this novel host plant might affect the interaction of these caterpillars with this pathogen. And what we found was really interesting. So this is a graph of survival to the adult stage. And what I'm showing here is survival when either there was no virus detected in the adults or when there was virus detected in the adults. And the results were so cool. When there was no virus, survival to the adult stage was the same, whether caterpillars were reared on the introduced plant or the native plant. But when the caterpillars were infected, their survival to the adult stage was about 90% on the introduced plant and only 30% on the native plant. So that suggests, so this novel host plant then is allowing protection from this virus. And we suspect that it's the fact that they're able to sequester these iridoid glycosides from the introduced plant, from English plantain, that is allowing them to survive infection by this virus. So this novel host plant can provide protection against a viral pathogen. So what I hope I've been able to show you with these examples is that in natural systems, novel host plants can have both positive as well as sometimes negative effects. So overall, what I hope I've been able to do today is give you a glimpse into the fact that introduced organisms are not necessarily bad. They can be really, really bad. They can be serious problems, especially if you start talking about pathogens, but I didn't want to get into that. So even just thinking about insects and plants, these exotic plants may become invasive, may become problems, but they can also provide many benefits to the organisms with which they interact, including to we humans. And with that, I'd like to acknowledge my funding sources and the many, many people who've helped with the work that I've talked about today, and thank all of you for attending. Thanks so much.
Wow, is all I can say. I'm fascinated by this research. I want to invite, and I'm seeing some claps and virtual cheering for you for this great work and presentation. So I would love to invite our attendees to provide feedback, some comments, some questions. I'm happy to monitor both the chat and raised hands.
Yeah, I'm going to stop sharing my screen now.
Yep.
And I'm happy to answer any questions.
We're getting some great feedback in the chat. So as people are thinking, I'm really curious, how did you get started in this line of applied work and research?
Well, it actually started when I was 7 years old and began collecting butterflies.
Oh.
And then I got really interested in, unpalatability as a feature of many insects, and did some of my graduate work in the lab of Lincoln Brower, who did all the classic work on monarch unpalatability. And so I just have became fascinated by the ability of insects to deal with this amazing diversity of chemical compounds that occur in plants. And so all plants on the planet contain chemical compounds, and some of these are, really toxic. Think about, strychnine. It occurs in plants. But insects can deal with those compounds, and so I became really fascinated with that whole phenomenon and how it works, and then how the host plant that an insect feeds on can moderate those interactions.
It's fascinating work. I do want to note that you're getting some great feedback in the chat. A couple I'm happy to read to you. Johanna, very, very interesting. I had no idea introduced species, whether purposeful or not, could be good or have good effects like that. Giorgio, I'm sorry if I'm not saying the name accurately, did have a question. Very interesting stuff. Did the white peacock have a predisposition for sequestering plant toxins?
Right. That was a new phenomenon. Nobody had ever really looked at that in the species before. We knew that other species that had fed on English plantain could sequester iridoid glycosides from it. So we decided we wanted to look at it in this species too.
Wonderful. Kumiko asks, does this plantain provide both viral protection? Does the plantain provide viral protection for both butterflies? Great topic.
So because Taylor's checker spot is an endangered species, working with it is very difficult. And so doing the kind of experiments with this viral pathogen that we've done with other species would be really hard. So we haven't looked at it for that. We have looked at some other species, and it turns out that the plantain, the relationship is a little more complicated with other species. And again, it has to do with the chemistry and what specific chemical compounds are found in the plantain compared to other host plants. So we've worked with a couple of different species that I showed you in that slide there to look at viral protection. So sometimes plantain is good, and sometimes it's not as good as the native host plant, it turns out.
It depends. Wonderful. We have a question from Suzanne. Although becoming newly unpalatable to other species, is it beneficial to the caterpillars that feed on the plantain? Is there any information on how that may affect the bird populations that largely depend on caterpillars for feeding their chicks?
Okay, that's a great question. So a lot of birds are really smart. And so they learn which species are unpalatable, and they don't feed those to their young.
Fascinating. Raul has a question. He says, I know this is a bit out of topic, but I would like to hear your opinion regarding the decline of insects caused by human activities and what we may expect in the future.
So, well, certain groups of insects are declining. There's no question about that. Not all groups of insects are declining. I don't think it can quite be called the insect apocalypse, but for certain groups, bumblebees are a big group where we're seeing really major declines in bumblebee abundance. And what causes it? Too many people, urbanization, too much use of pesticides, lots of different things have negative effects on insect populations.
Thank you for that response. Betsy says, could we do a better job detecting and preventing invasion by other foreign species? I wrote a paper on an invasive nematode in New York forests that we didn't know we had, that China notified us about after they found it in lumber from the US.
Oh, that's interesting. I mean, basically things sneak in. And so it's thought that the spotted lanternfly came in from an egg mass that was on, one little egg mass that was on a shipment that came over that just wasn't detected. And so there's a lot of inspection. If you fly internationally, you have to go through, you can't bring certain foods in and stuff like that. So yes, we could probably do a better job, but because people travel so much, you can get egg masses on the underside of your car, of problematic species. You can get, if you collect wood for a campfire in one state, you could move a problematic species from one state to another. So yes, we could do a lot better job, but we as individuals have to be careful as well.
Wonderful. I do want to recognize that I'm getting a lot of gratitude and appreciation coming your way for your great work and this presentation. Betsy follows up and says, but China is inspecting lumber. Think of all the shipping containers.
Yeah,
absolutely. Wonderful. We're caught up in the chat. I do want to pause and offer an opportunity for people to add to the chat or even raise your hand. Fabulous talk, great feedback, lots of thank yous coming in. As you're thinking about final thoughts, I do want to put in the chat a couple of links. We are offering Let's See You Well talks on a regular basis, so you can check in on the Be Well site and look for Let's See You Well. And I've also entered a feedback form if you'd like to take just a couple of minutes to give your feedback for this most excellent presentation. Again, lots of thank yous coming through. I would like to do a deep bow of gratitude to all of my Be Well partners for helping to produce this content. Certainly all of the participants for your great interest in showing up, but most importantly, thank you, Dr. Deane Bowers, for your great insights, research, presentation. You're helping to make our community more well, and we are most grateful.
Thanks so much, everybody.
The recording will be posted on the website when our comm folks get a chance, and thank you all for coming. I hope you're seeing all the love they're giving you in the chat, Deane.
Yes, I am. Thank you.
All right, awesome. Well, I'll sign off then for the group. Thank you, everyone. We'll see you at the next one.
Thanks so much, Erin. I really appreciate it.
My pleasure. We're very grateful.
All right. Bye-bye.